Let's say there's a gene in zebrafish that causes suppression of cancer. We'll call it FAB. We would like to see whether decreased or zero expression of this gene is associated with development of particular types of tumors. To do this, we can use RNA interference, known as RNAi, or clustered regularly interspaced short palindromic repeats, known as CRISPR. Let's go back to the central dogma of genetics. DNA gets replicated, transcribed to RNA, and then translated to proteins. Now let's talk about RNA interference. First, double-stranded RNA, or dsRNA, is introduced into the cell. After dsRNA has been taken up by the cell, dicer, which is an RNase enzyme, cleaves the dsRNA into fragments, which are called small interfering RNAs, or siRNAs. Our siRNA will then separate into two strands, and the antisense strand binds to the RNA-induced silencing complex, or RISC. And now, siRNAs and the RISC complex bind to target homologous mRNA. The target mRNA, in our case FAB, gets cleaved and can no longer be translated, so we have knocked down or reduced expression of this gene. In CRISPR, an RNA guide molecule is programmed to match a unique DNA sequence, in our case FAB, and then the RNA guide attaches to an enzyme called Cas9, which is found in many species of bacteria and is a natural source of immunity, in order to target our specific DNA sequence. The RNA aligns with the target DNA sequence, and Cas9 locks onto and cuts both strands of the matching DNA. The DNA cut can be repaired with an insertion of DNA or by joining the broken DNA segments together, creating a frameshift mutation. The end result is that we have disrupted gene expression on the genomic level rather than simply disrupting RNA expression. So with CRISPR, we obviously have more precise editing capability, and we're able to completely stop expression of a gene, knocking it out. RNAi can only be used for gene silencing, while variations of CRISPR can be used for gene knockdown, knockout, activation, suppression, or base editing, so it's a lot more flexible. Disadvantages include possible off-target effects, which would be nonspecific and unintended genetic modifications. CRISPR is also more complex to actually do in the lab than RNAi, and because you're completely knocking out a gene, that gene is vital, you're actually going to cause cell death. With RNAi, we see similar advantages. Incomplete reduction of gene expression or knockdown of a gene allows you to test different levels of expression. You can also reverse RNAi in some situations or rescue your target cells. We have been doing RNAi for longer. It's a well-established procedure, so that is another advantage. It's not as specific as CRISPR, so you're going to see higher rates of off-target effects, and it's obviously not as versatile. So which technique is better? It depends on your experiment, but in my hypothetical situation, I would go with CRISPR because complete knockout of a gene will give us a more pronounced phenotype and will allow us to more readily see how FAB suppresses cancer.